Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 15 from our Brock Biology of Microorganisms textbook. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, let's get started. Chapter 15 covering functional diversity of microorganisms. We're trying to make sense of microbial diversity in the first part of the chapter here. Now, there is such thing as phylogenetic diversity. This means evolutionary relationships between organisms, where you're comparing organisms based on relatedness and looking at taxonomy. And then there's functional diversity, where you're looking at form and function as related to microbial physiology and ecology. So this chapter focuses on functional diversity over phylogenetic diversity. We're looking at functions of different microorganisms and how these functions can be distinct to different groups. Here you can see major phyla. These are different phyla of prokaryotes. In red, you have the archaea, and in blue, you have the bacteria. And notice that this is a nice little diagram depicting different functions that the various phyla of bacteria and archaea possess. Now, what are some reasons behind having functional traits in different species and different phyla? You see how different phyla may have some of the same functionality? What is the reason for this? Well, one major reason is convergent evolution, where a trait has evolved independently in two or more lineages, but is not encoded by homologous genes. You can think of it as the wings of a beetle versus the wings of a bird, right? They're both wings, but they evolved independently of one another. They aren't due to lineages and evolution. Then there's gene loss. A trait can be present in a common ancestor, but was later lost. That ability may have been lost, or that function may have been lost. And certain groups of microorganisms can acquire functions by horizontal gene transfer. Remember, we talked about a form of horizontal gene transfer called, with the sex pilus, conjugation that occurs. Genes that encode for a trait can be exchanged between distantly related lineages. Remember, so for example, during conjugation, the donor organism can give genetic information to the recipient, even if it's a different species. This is just making sense of some of these functions and how different functions can exist in seemingly distantly related phyla. Now, let's move on to the next part of the chapter, looking at diversity of the phototropic bacteria. Phototropic meaning getting their energy from the sun, from light. Now, what is an overview of these phototropic bacteria? Remember, the Earth was anoxic. There was no oxygen in the atmosphere. And the first phototrophs were anoxygenic phototrophs. This means that they did not generate oxygen as a product. Remember, organisms that undergo phototrophy that do not produce oxygen as a product, this means that they do not use water as their electron source, okay? Those phototrophs that produce oxygen as a product use water for photosynthesis as an electron source. Those that do not produce oxygen do not use water as their electron source. So that's a good shortcut, cheat sheet way of remembering the difference between the two. Now, let's talk about cyanobacteria, the most important, in my opinion, of these prokaryotic photosynthesizers. These are oxygen producing phototrophs. They are so diverse. They're large, diverse in their morphology, ecologically heterogeneous group of oxygenic, that means they produce oxygen, phototropic bacteria. Remember, phototropic means they get their energy from sunlight, they produce oxygen here. And this is one of those groups of microorganisms that's very important to life on Earth because they produce oxygen for us to breathe, right? And they are a huge contributor to oxygen production on Earth. And they undergo your classic photosynthesis. Like I said, they're morphologically diverse. They can be unicellular, they can be filamentous. There's so many different types of these cyanobacteria. They can be smaller in size, very large in size. And like I said, they can undergo photosynthesis. But remember, these are prokaryotes, right? Cyanobacteria are bacteria. They do not possess chloroplasts. However, they do have thylakoids. Remember those pancakes when we talk about granum inside of the chloroplast? You have those stacks that look like pancake stacks, right? A granum is a stack of thylakoids. Well, these bacterium do have thylakoids, and they do have a thylakoid membrane, but it is an extension of the plasma membrane, okay? They have thylakoids as an extension of the plasma membrane. Here you can see the thylakoids of cyanobacteria, and you can see they kind of look like pancake stacks in here, but they are stacks inside of the cell itself in the cytoplasm, and they are an extension of the plasma membrane. Now, moving on to what are known as the purple sulfur bacteria. They are purple because of their carotenoids. These are special pigments that they use, for photosynthesis, for their type of phototrophy, I should say. They are found in anoxic zones. They do not produce oxygen. They are anoxygenic phototrophs. And remember, what does that mean? If they don't produce oxygen, that means they don't use water as their electron donor, as their electron source. So let's look at what the purple sulfur bacteria use as their electron source. They use hydrogen sulfide, H2S, as their electron source, as their electron donor for photosynthesis. So they're using sunlight for energy, and then they're taking those electrons, see these hydrogens? They're taking these electrons from hydrogen sulfide, and that produces sulfur, right? And that's why they're known as the sulfur bacteria. They're producing sulfur because they oxidize hydrogen sulfide as an electron source. Isn't that interesting? So these do not require water as the electron source, and they do not produce oxygen because they're not oxidizing water to oxygen. Instead, they are producing sulfur because they are oxidizing hydrogen sulfide to sulfur. Very interesting. And where do we find these guys? We find these guys in microbial mats and salt marsh sediments. Here you can see some of these blooms of purple sulfur bacteria. Here in A, you can see them in a sulfide spring. Here you can see their purple, beautiful purple color. And here you can see them up close under a phase contrast microscope. This is a micrograph that you can see the bacterium here. All right, moving on to another functional type. These are known as the purple non-sulfur bacteria. Here are a list of key genera. These are highly metabolically versatile. They are very interesting. They have a purple, red, orange color. You can see some of their colors here. They range from purpley to blue to yellow, red, orange. And they are typically photoheterotrophs. This means that they get their energy from sunlight, and remember, they get their carbon from an organic source. Very interesting, right? So they use a wide range of carbon sources and electron donors for photosynthesis. For example, they can use organic acids, amino acids, alcohols, sugars, aromatic compounds. That's where they get their carbon from. And that's where they can get their electrons from too. So they are anoxygenic, right? They don't produce oxygen because they don't get their electrons from water. Here you can see some of these representatives under the microscope, and see how morphologically diverse they are. There are many different forms, many different shapes of these bacteria. Now, moving on to the aerobic anoxygenic phototrophs. Aerobic anoxygenic phototrophs. Here's some key genre. These are obligate aerobic heterotrophs that use light, right? They're phototrophs. They use light as a supplemental energy source. They don't need the energy from the light, but they can use it for supplemental energy. They are strict heterotrophs. That means they get their carbon from organic molecules, and they employ anoxygenic photosynthesis. So this means that they do not produce oxygen. So they're not using water as an electron source. And they are unable to fix CO2 because they need organic carbon. Their carotenoids provide a yellow color, an orange color, even a pink color. And they perform 5% of the photosynthesis in coastal marine waters. Very interesting. Now moving on to the green sulfur bacteria. Here are some key genre. This group is able to oxidize hydrogen sulfide, H2S, to sulfate. This group is strictly anaerobic and anoxygenic, which means they do not require oxygen for growth, and they do not produce oxygen either. And lastly, for the phototrophs, let's discuss the green non-sulfur bacteria. Here are some key genre. And by the way, when I say here are some key genre, you don't have to memorize these key genre for my class. But these are the different genre who are considered to be functionally green non-sulfur bacterium. They tend to be filamentous, anoxygenic phototrophs. So filamentous in their shape, their morphology. Anoxygenic, meaning they do not produce oxygen. So again, what does that mean? They don't use water as an electron source, but they are phototrophs. They do get their energy from sunlight. In this class in particular, in this phylum in particular, Chloroflexi, the Chloroflexi are green non-sulfur bacterium. Now, before we leave phototrophy and talk about some of these other functional types, sulfate-reducers, sulfur-reducers, sulfur-oxidizers, before we move on to that, let's take a quick break time with Gizmo and Wicket, my cats, and we'll be right back with this latter half of the chapter. All right, welcome back from break time with Gizmo and Wicket. Let's carry on with our discussion of functional types of microorganisms. We're going to start our discussion here with dissimilative sulfate-reducers. Here's some key genre. And what we need to know about these is that they couple oxidation of hydrogen gas or organics to reduction of sulfate. This is a form of anaerobic respiration. So let's break this down. They use hydrogen as an electron donor, as an electron source. And those electrons are picked up by the terminal electron acceptor sulfate. So they reduce sulfate. So if you were to guess, do these require oxygen or not? Are they anaerobes or aerobes? They are anaerobic bacteria. They do not use oxygen as a terminal electron acceptor like you and I do, okay? They do not use oxygen. So they do not breathe oxygen. They actually breathe sulfate. Isn't that interesting? They take electrons from hydrogen gas. That's where they get their electrons from. So they are lithotrophs, and they reduce sulfate. So they undergo a type of anaerobic respiration. So here you can see some of the different groups of sulfate reducers. Next, let's talk about the sulfur reducers. The sulfur reducers. Here are some key genera. These use respiratory reduction of sulfur to hydrogen sulfide. Now, what does that mean? Reduction. Reduction means that they give their electrons to sulfur. Sulfur is the terminal electron acceptor. Sulfur picks up the electrons at the end of the electron transport chain. So sulfur reducers breathe sulfur. They use sulfur as a terminal electron acceptor at the end of the electron transport chain. Sulfur then gets reduced to hydrogen sulfide. So they produce hydrogen sulfide. They do not require oxygen. They do not breathe oxygen. You're going to find them in sulfur springs, and you're going to find examples of these types of bacteria, and you're going to find examples as archaea as well. Now, moving on to the sulfur-oxidizers. Here are some key genera. These are chemolithoautotrophs that oxidize, I think it's spelled wrong here in your notes, that oxidize hydrogen sulfide, sulfur, thiosulfate, and thiocyanate. So that means that they get their electrons from these molecules, okay? They get their electrons from these molecules. That's what oxidizer means, right? If an organism is a sulfur oxidizer, it means it gets its electrons from a sulfur molecule. Okay, where do you find these? Obviously, you're going to find them anywhere there's rich sulfur, like sulfur springs, marine sediments, hydrothermal systems where H2S is produced by other bacterium, or geothermal reactions. Anywhere is rich in sulfur, you're going to find sulfur oxidizers. And again, there are 3 phyla of bacteria that are sulfur oxidizer, and 1 phyla of archaea, which is a sulfur oxidizer. Okay, and again, you're going to find these sulfur oxidizers in places like this sulfidic artisan spring in Florida. You can find them in these chimneys, these hydrothermal chimneys at Cathedral Hill in Guaymas Basin, I should say, in Mexico. You're going to find these types of sulfur oxidizers any place where you can find rich sulfur springs, sulfur vents. Now, moving on to nitrogen using bacterium. Okay, we're going to look at nitrogen fixers, and then we're going to look at nitrifiers and denitrifiers. So what are nitrogen fixers? Here's some key genera that are able to fix nitrogen. Nitrogen fixation means the ability to take nitrogen out of the air, right? Most of what you're breathing right now is nitrogen. Most of what is in the atmosphere is nitrogen. These organisms are able to fix nitrogen. They're able to take nitrogen out of the air, and these diazotrophs, they can take nitrogen out of the air and fix it into ammonia. So they're able to essentially convert nitrogen to ammonia. Nitrogen fixation is widespread among bacteria, and a few archaea are able to do this as well. Now, moving on to the difference between nitrifiers and denitrifiers. Denitrifiers grow by anaerobic respiration of some form of inorganic nitrogen, either nitrate, NO3, or nitrite, NO2. They are able to, what does anaerobic respiration mean? Anaerobic respiration means that you use something other than oxygen as your terminal electron acceptor. So these denitrifiers use nitrate, NO3, or nitrite, NO2, as their terminal electron acceptor at the end of cellular respiration. That means that they reduce NO3 or NO2 to what? To gaseous nitrous oxide, nitrogen dioxide, dinitrogen oxide, and nitrogen gas. By the way, this is why they are called denitrifiers. They remove nitrates and nitrites from soil, right? They convert nitrates and nitrites to gaseous forms. And so they denitrify. They remove nitrogen from the soil. They remove nitrogen, right? So if you're like a farmer, these are not your friends, right? You don't want organisms in the soil that are removing those rich these are examples of nitrogen molecules, but those rich nutrients, you could think of them as, from the soil. These organisms are removing those nitrates and nitrites that your plants like in order to grow from the soil. So if denitrifiers remove nitrate and nitrite from the soil, what do you think nitrifiers do? The opposite, right? So these grow chemolithotrophically at the expense of reduced inorganic nitrogen compounds. So these ones grow by using ammonia and nitrite as an electron source, right? So they are able to oxidize ammonia and oxidize nitrite. And when they oxidize ammonia or nitrite, they actually produce nitrite and nitrate. So if you're a farmer, nitrifiers are your friend. They are actually helping to provide the soil with nitrates and nitrites, right? By converting ammonia to nitrite and converting nitrite to nitrate. So here we can see some phase contrast micrographs of nitrifying bacteria. And moving on, let's move on to functional types that involve iron, methane, and other types of functional groups later on as well. So what are these functional groups here? These are called the Dissimilative Iron-Reducers. What does reduce mean? So always break it down at the beginning, right? So what is an iron reducer? It means that you give electrons to iron, right? Reduce, remember Leo says Gur, loss of electron means oxidize, gain of electron means reduced, right? So if you're an iron reducer, you're giving electrons to iron. The only place where you can give electrons is at the end of the electron transport chain. That means that these organisms breathe iron, right? So they use iron as a electron dump. It's as a terminal electron acceptor. So they carry out, get this, they carry out metal respiration. That sounds awesome. They use metal, they use iron as their terminal electron acceptor. So they couple oxidation of hydrogen gas. That's where they get their electrons from. They get their electrons from hydrogen gas, or they can use organics. And then they reduce ferric iron, so Fe3+. They use ferric iron as their terminal electron acceptor. Some can even use manganese, Mn6+, as their terminal electron acceptor. So this is a form of anaerobic respiration, metal respiration. Now, what about dissimilative iron oxidizers? What does that mean? Oxidize. Oxidize means to take electrons from, right? So these guys get their electrons from iron. So they are lithotrophs. They get their electrons from an inorganic source, in this case, iron. They eat iron, okay? These are iron eaters. So here's some key genre. They couple iron, Fe2, iron, to cell growth. They take electrons from iron, right? They eat iron as an electron source for the electron transport chain. Aerobic iron oxidizers, diversity and distribution influenced by pH and O2. So some of them can be aerobic. That means that they use oxygen as a terminal electron acceptor, but where do they get their electrons from? From iron. Isn't that interesting? Now, moving on to methane producers or methane users, let's talk about that. So what are the Methylotrophs? Methylotrophs. The Methylotrophs grow using organic compounds lacking carbon-to-carbon bonds. So here's an example of a methylotroph. Mehanotrophs, right? Mehanotrophs are a subset of methylotrophs that use methane for growth. Remember, methane is CH4. These methanotrophs, which are an example of methylotrophs, these use methane as an electron source for growth. Okay? Now, moving on to another functional type. These are predators, okay? These are microbial predators. They get their nutrition from consuming, from eating other organisms. Here's some couple of genera, and these bacteria consume others. So for example, Delovibrio are periplasmic predators that invade and replicate within the periplasm. So think of almost like the life cycle of a virus. The Delovibrio are able to invade an adjacent bacteria to get into the bacteria. They then grow in length. They undergo a process known as multiple fission. They then break out of that cell and then invade more cells. So that's an example. Delovibrio is an example of a predatory microbial organism that gets its nutrition by literally behaving like a virus, getting into bacterium, growing within there, getting the nutrients it needs from that bacterium, and then exploding the cell and getting out and, doing it over again. Now, the final group here, the bioluminescent microbes. These functional types can bioluminesce. That means that they can emit light, right? They can emit light. Here's some key genre. Here's some key genre here. Now, these are mostly marine. Okay, these are mostly marine. Some of them localize light in organs of fish and squid. They can produce light for signaling, avoiding predators, affecting prey. So oftentimes they are in symbiotic relationship with marine life, with fish, with squid, and their bioluminescence helps the fish, helps the squid in order to hunt or avoid prey. And in return, these bacterium, they get to, get a source of nutrients when the fish or the squid feeds. So here you can see these bioluminescent bacteria, and you can see them. They can localize to, structures in the face of the fish. You could see them on squid structures as well. Here they are growing on a plate. You can see that the colonies themselves glow. Isn't that interesting? But only when they reach sufficient concentrations. Remember when we learned about quorum sensing? These bacteria will not glow. They will not bioluminesce until they reach sufficient concentrations. Okay, so we reached the end of the chapter. Good job, you guys. Pretty short chapter, but very interesting, touching on the different functional types of bacteria. I hope you learned a lot. Let me know if you have any questions in the comment box below, and I'll catch you guys for chapter 16.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.